Hello, greetings from Vienna. My name is Alexander Haselberger. I am from University of Vienna, and I would like to discuss with you some aspects of aging and functional foods. I'm sitting here in front of the castle of Schönbrunn, and you can see here also Empress Elizabeth. She lived here, and she was famous for her ideas and fancy ideas also to stay healthy, young, and beautiful. So let's see if we improved since then. I would like to go with you through some aspects of aging mechanisms, epigenetic, senescence, and present some experience from a recent study of us. The work was done in cooperation with HBC and Blue Zones. I think we all know this pic here, showing the hallmarks of aging and main mechanisms such as mitochondrial dysfunction, senescence, DNA instability, epigenetics, and more. And we also learned about the mechanisms of epigenetics, DNA methylation, histone modification, and non-coding RNAs, regulating gene expression and DNA stability. We even learned from the Howard clock that epigenetic methylation can tell us if lifestyle, stress, and nutrition drives us toward accelerating aging or slowed down healthy aging. Epigenetic even regulates many of the mechanisms of hallmarks, like microRNAs, histone acetylation. It affects telomeres, mitochondrial biochemical pathways, the single steps of autophagy, or different mechanisms of DNA repair. We reported recently that epigenetics is also involved in the mechanisms of infection of cells by COVID-19 and in the way functional foods can interfere with these mechanisms and potentially protect against infection. Another central element of aging are our commensals, or microbiota. They change in structure and amount, or bacterial groups where an inflammatory prone species become more, where diversity goes down and production of anti-inflammatory short-chain fatty acids also go down. A number of polyphenols and especially also their metabolites have been shown to affect microbiota structure positively. And especially also microbiota metabolites such as short-chain fatty acids can be regulated by functional foods. In the last years, it was shown very clearly that main elements in our body, the brain, the immune system, the microbiota gut, and the endocrine system are exchanging information and are interfering, such building an axis which participates a lot to our personal health and aging characteristics. Environmental stress factors like social stress, radicals, or toxins, we are just now working on bisphenols in this area, are disturbing almost all epigenetic mechanisms, resulting in impaired physiological functions, in accelerated aging, or in age-related diseases, especially of the immune system, or even in cancerogenesis. Recently, we learned about a special effect of radicals, mediating changes in the DNA formation, the building of quadruplexes at guanines. They affect DNA methyltransferases. They can affect or result in mismethylation. They can affect telomeres and obviously many other mechanisms. Radicals, DNA damage, telomere erosion, and epigenetic changes are also involved in the formation of senescent cells in our tissues. If they are getting too many, this leads to dysfunction, tissue destabilization, chronic inflammation, and potentially cancerogenesis. You can see here the typical beta-gal blue colored senescent cells. The senescence associated secretory phenotype with its many inflammatory proteins and damaging, including insulin resistance. So what can we do against these hazards of a healthy aging? And the good, already well-known news is that caloric restriction such as fasting periods or intermittent fasting, activate molecular mechanisms which counteract many of these dangerous effects. This especially includes activation of Sirt proteins, p53 and PGC1 alpha pathways. And we also think that a number of polyphenols activate similar pathways, such mimicking caloric restriction. To compare effects of caloric restriction and polyphenols was the effect of the objective of a study of our group. Especially the modulation of Sirts and the consequences in vitro and vivo was part of our project. Sirt1, Sirt3 and Sirt6 and others are well known for the molecular effects of mitochondria, elements of the circadian clock, on epigenetic mechanisms, especially histones, and their effects against inflammation or senescence, and also for help of DNA repair mechanisms. Since a few years, the idea of senolysis, the eradication of senescent cells from one or more tissues, became vibrant, and names of numbers of drugs, but also of plant ingredients or functional foods have dropped. This is certainly of major health and economic interest. We studied epigallocatechin-galate, EGCG, resveratrol, anthocyans, spermidine, fluoridine, but also short-chain fatty acids, butyrate and beta-hydroxybutyrate in a model of pre-adipocytes and adipocytes. Very shortly, 3T3L1 cells were incubated for 8 days with BRDU and 4 days including the test compounds in various concentrations. Beta-gal staining in the picture shows senescent cells. Or 40 days incubation or 14 days for differentiation with growth mixes, and then you can see fat droplets in these cells and analysis of markers. We looked then for the cultures and molecular markers. This work is already submitted for review. You can see here in pre-adipocytes that epigallocatechin-galate stimulates Sirt3 significantly. Tested polyphenols in the picture shown is EGCG modulate CDKN1 alpha, which is P21, a marker for senescence. In adipocytes, preliminary data indicate that butyrate and spermidine, but also EGCG modulate the expression of ATG7, a protein involved in autophagy. And we see opposite effects on PPAR-gamma expression, a mechanism in differentiation by butyrates, spermidine, but also EGCG and fluoridine. In a human study, we compared effects of fasting and a drink containing mainly Sirt-activating ingredients. The fasting study involved 20 people, including me, and fasting was according to the Buchinger method, including colonic irrigation in the beginning, 5 days, 150 calories, and drinking a lot and doing a lot of exercise. The third study involved 103 participants and a control. The participants took a drink containing anthocyanidine, fluoridine, sulforaphane, epigallocatechin-galate, allopaein, nicotinamide riboside, vitamin C over 3 months. Markers from capillary blood were then analyzed in blood spots, where these blood spots are really stable and a nice tool for analysis. We found, as expected, increased levels of acetate and highly significant levels of butyrate. I would not know if intermittent fasting really comes to similar levels of butyrate. And butyrate is of course well known for its anti-inflammatory and epigenetic modulating effect in metabolic syndrome, in neurological diseases, and many more. Both fasting and Sirt intervention increased the expression of Sirt1 and microRNA 34. This microRNA 34 is well known for its activity in combination with SIRT1, with the SIRT1 and p53 pathway. Pyruvate dehydrogenase, DDK4, is an important marker in glucose, glycolysis, and fat metabolism, also involving activities in mitochondria. We interestingly found increased expression of PDK4 after fasting, as well after the Sirt intervention. Effects of polyphenols on microbiota are well known, and already a few years ago we reported that fasting and EGCG improves microbiota structure, especially an increase in diversity and an increase of short-chain fatty acid producers. Also in this study, Illumina sequencing reveals that after fasting, you can see at T2, and fasting plus a Sirt intervention, T3, at phylum level, actinobacteria and verrucomicrobia increased. And this is mainly due to an increase in bifidobacteria and Akkermansia. And here you can see the increase of actinobacteria, especially bifidobacteria, after Sirt intervention only. And again, the present study, we found an enhanced expression of MLH1 after fasting and after Sirt intervention. And from all these aging effects, I personally would consider DNA repair as the most important effect. Of course, there are more DNA repair mechanisms which need to be investigated. We published a number of results in this area. You can see the publications on the side of this slide. MicroRNA 16 is an exciting guy. It's active in the regulation of cell cycle, and maybe because of this, it has also been reported for the control and repair of the gut barrier cells, obviously interacting with tight junctions. We found this microRNA enhanced following fasting and Sirt intervention. Well, coming to summary and conclusions. Of course, there are probably 10,000 of molecules, pathways, and markers which need to be analyzed and understood as a consequence of such fundamental interventions, such as fasting and complex interventions with Sirt foods. We analyzed a set of markers of targeted mechanisms in blood spot. We do find striking similarities between effects which are postulated as a result of fasting and Sirt activation as a reaction of fasting, as well as following an intervention with a drink containing Sirt activating ingredients, a functional food. Let me add my conviction that this research field adds to an important process of healthcare, which is going on nowadays. In contrast to conventional healthcare, which starts intervention after the appearance of disease symptoms, in future, marker will allow us to make interventions before even symptoms establish. However, the marker need to be really good markers. But this area will rapidly result into preventive and personalized disease prevention and health preservation. So it will go away more from the thinking of a pathogenesis, going towards a thinking of salutogenesis, aiming for health. And last not least, in the crazy field of aging and rejuvenation, what are the real central objectives? I think we should really rethink these objectives. Is it really the increase of longevity and lifespan of a few? Is it fancy for media, or is it important for health? Or is it more important to increase healthy lifespan and the prevention of aging-related diseases? At the end, please let me thank my group, especially Stefanie Lilja and Julia Oldenburg, You can find information about my lab under my website alexander/haselberger.at, and you can find information about personal health thinking under this website on the left-hand side, and more information about our partners, healthbiocare.com and Blue Zones. Thank you for your interest.